Hi, everyone. My name is Nicholas Verhoeven. I'm a PhD student in molecular medicine, and I am also a cell biology researcher at my medical school. And today we're going to be discussing a topic that was actually, I was going through the Joe Rogan Experience podcast, and I always really enjoy, obviously, I specifically like to listen to his medical podcast where he has some sort of expert on that talks about their particular expertise, but specifically in medicine, or if that's cell biology or molecular biology or anything in kind of that realm. And maybe like a week ago, he had a podcast where he discussed neuroinflammation and how that was related to PTSD, how that was related to TBI, so traumatic brain injury. And this person, Dr. Mark Gordon, I believe is his name, briefly mentioned a chemokine, a cytokine, a molecule, those are all kind of the same thing, known as fractalkine. And that kind of piqued my interest, so I decided to do a little bit of digging. So I've done some reading on this particular chemokine, and I'd like to kind of lay out the information that I found out on this particular topic. Before I get started, I'd like to thank everybody that did end up buying the creatine ebook that I've released. It certainly meant a lot to me. It's not like I expect a ton of money out of this stuff, but it's certainly impactful to me to know that I've got a community of people that really care about learning a science-based education. So thank you to those that have supported me in that way, and hopefully the book is coming out to be useful for you. But with that out of the way, let's go ahead and jump into this fractalkine cytokine. And I'm going to go into a pretty decent amount of detail based off of a review that I went through, and I actually ended up reading a few different studies on this, but this particular review is from the Annals of New York Academy of Sciences, which is fractalkine in the nervous system, neuroprotection or neurotoxic molecule. So there's going to be a little bit of nuance here that wasn't necessarily explained in the Joe Rogan podcast if you ended up listening to that. So if you are just listening to the podcast, as usual, that's going to be okay. I'm going to describe things as we go along. But if you would like some visuals, then I'm going to have a few visuals that pop up on the screen for anybody who's actually watching the podcast, as that is in certain ways has its advantages. Okay, so let's dive in. Fractalkine, which is also known as, and I'm definitely not calling this this going forward, but CX3CL1, that's a fun name. I'm going to go with fractalkine. So fractalkine is a chemokine. So it has a series of different effects, but chemokines as a general category typically attract other cells. So they allow other cells, they kind of convince other cells to migrate to a particular local area. And that's obviously important when it comes to inflammation. If you want to attract inflammatory cells, as we'll see, those can then move to a particular localized region. Now, what was discussed by Dr. Mark Gordon, and I'm not at all saying that what he said is correct or incorrect, mainly because I have not vetted this enough, but I did want to go into some detail on this particular cytokine or chemokine. But what he mentioned is that in people who have PTSD or they have a traumatic brain injury, that this fractalkine protein will disappear out of the brain. So if they're healthy, everything is fine, fractalkine is present. And then when they have some sort of pathology that occurs, fractalkine just mysteriously disappears. And I'm going to explain a little bit of why that might be the case, as well as a pushback against that idea a little bit as well with my limited reading that I've done. So where does fractalkine come from? Well, it comes from neurons. So these neurons actually produce this protein, and this protein is actually expressed as what's called a transmembrane protein. So transmembrane meaning that it's anchored in the actual membrane itself, and then what happens is you have another protein known as a protease, And that protease will come by, and on the outside of the cell, the part of the fractalkine protein that is exposed to the outside of the cell will then be cut by this protease. Now, there are a few different proteases that act on this particular protein. There's a CAT-S and an MMP, also known as ATOM protease. Those are particular names. You don't actually have to know those, but just know that a protease, which is a general class of protein that specifically cuts and cleaves proteins, other proteins, ends up releasing fractalkine into the liquid, into essentially the cerebrospinal fluid, and in the fluid around the brain. So you have one version that's bound, the fractalkine that's bound to the neuron, and then you have another version that's soluble or cleaved, and that is released by that protease. So what typically then ends up happening after it gets cleaved is you have its effect on, and let me see if I can find this visual right here. What typically ends up happening, this cleaved fractalkine will then bind what's known as a GPCR. So G protein coupled receptor. And this fractalkine receptor will then lead to a series of changes, but specifically changes in a type of brain cell known as a microglia. Microglial cells are essentially immune cells that are specific to the central nervous system. And in this case, we're more focused on the brain itself. So it specifically binds microglial cells, and we'll exactly see why it does that. I'll explain that in just a little bit. But just going a little bit more into this background. So this G protein is, or this receptor is also called CX3CR1. Clearly, I'm not going to be calling it CX3CR1 from now on. Just know it's the fractalkine receptor, and fractalkine binds to it, and then it leads to a change, and we'll discuss that change in just a little bit. But generally, fractalkine regulates synaptic plasticity and cognitive function by depressing excitatory postsynaptic potentials, EPSPs. What does that mean? Well, when our brain is firing, let's say it's trying to communicate from one neuron to the next neuron, what it can use is a neurotransmitter known as glutamate, which is the most common form of neurotransmitter in the brain. And what can happen is if you have an overabundance of this glutamate, which can happen in a variety of different pathologies, if you have an overabundance of this glutamate, that can lead to an overexcitation of the nearby neurons. And what that can do is actually damage those neurons. If you do too much of this excitation, that goes through this molecule, glutamate, that binds glutamate receptors, and then has its own signaling cascade within the cell, which then tells that cell to change in particular ways. I'm not going to go into too much detail with that, but just know that glutamate tends to be excitatory. So what fractalkine does is it actually depresses that excitatory state, which is a benefit when you have an overabundance of this glutamate. And glutamate can be detrimental when you have pathologies, even something like actual physical trauma that's occurring to the brain, because the neurons end up having issues regulating this glutamate. So fractalkine reduces glutamate, and it also dephosphorylates glutamate receptors. So you can have different tags that get added to particular receptors, and that can change the functionality of that receptor. And in this case, we're talking about glutamate receptors. So fractalkine, once it binds to, let's say, the microglia, then it will lead to a cascade which will change a series of different things. And again, I'm going to go into those specifics in just a little bit, but ultimately that leads to a dephosphorylation of these glutamate receptors. And that can be on neurons as well as, I would imagine, also in microglia, but don't quote me on that. Finally, the last thing I'd like to quick mention before I go into more of the details is that fractalkine is actually highly expressed in young individuals and tends to be less so in older individuals. So that tells us that we have this potential molecule that could be useful in a series of brain disorders. So I'll be going into those as well. Okay, so now let's look at kind of the evidence for kind of its neuroprotection through fractalkine. And then we're going to go into a little bit on the neurotoxicity that's seen through fractalkine as well. Okay, so injured neurons, if that's through physical trauma or through some sort of chemical trauma, respond to, well, in this case, any sort of insult by releasing fractalkine through greater expression and cleavage to create that soluble form. So again, that soluble form is the one that ends up interacting with the microglia and then ends up causing some sort of change on the microglia. So it's believed to be protective in hypoxic and excitotoxic situations. What does that mean? Well, it's thought to be protective when you have a lack of oxygen delivery to the brain, which is called hypoxia. And it can also be excitotoxic, which is what I was talking about earlier with glutamate excitotoxicity. That tends to be through that mechanism, through an overabundance of glutamate or an overabundance of binding to the glutamate receptor, or an overabundance, or you've got too much of this glutamate that's latching onto the receptor and it's not being sequestered off of the receptor. It could be through a variety of different mechanisms, kind of a submolecular mechanisms, but ultimately the point is that glutamate can lead to excitotoxicity. And there's also evidence, which is actually something that Dr. Mark Gordon talked about in this podcast. And again, I just want to make sure, I don't know who Dr. Mark Gordon is really. So I don't know if he's a reliable source. I'm certainly not going to put my name behind that person without actually having backed more of what he, actually looked into a lot more of what he said. But he did mention that, as I mentioned at the beginning, that if you do some sort of trauma, some sort of damage to the brain, that ends up, you end up losing all this fractalkine. And what I found throughout my reading is that you can have greater levels of interleukin-1 beta, which is a cytokine that is specifically pro-inflammatory. So if you have greater levels of this interleukin-1 beta, that actually reduces fractalkine levels in the brain, which kind of falls in line with exactly what he was saying. So what ultimately that leads to is a decrease in the survival and proliferation of these neuronal cells, in these neurons. The proliferation would be becoming more neurons that you might want, or that might be useful for thinking and neural plasticity, the ability for the brain to adapt to different situations. So, and this actually also leads to a hyperactivity of the microglial cells. Again, those immune cells that I was just talking about, suggesting that fractalkine has an anti-inflammatory role. So that's one line of evidence for why fractalkine is thought to be a neuroinflammatory or anti-neuroinflammatory chemokine or cytokine. Again, those are the same thing. So it's thought that, and let me throw this image up here as well, it's thought that fractalkine, when it binds to the microglia, although they would typically be activated, so they can go from a kind of a quiescent state, or maybe not necessarily quiescent, but an anti-inflammatory state. So inflammatory cells or immune cells can be in an anti-inflammatory state or be in a pro-inflammatory state. And typically when they're more in that pro-inflammatory state, they tend to be activated. So these microglia would then, if they were activated, would then end up moving to the site of damage or the site of insult, whatever that might be. And what fractalkine does when it's cleaved from the neurons is it moves to the microglia, binds that GPCR or the fractalkine receptor, and the CX3CR1, if you'll remember that. And then what it does is it downregulates the activated microglia. So from going from a pro-inflammatory state, it actually suppresses that excitation of the microglia, making sure that they don't become overzealous or become overly active over time. So it's thought that fractalkine is constitutively released by the neurons. It's constitutively created, meaning that it's constantly created over time. So if you're healthy, if everything is fine, your neurons are constantly producing this fractalkine and constantly releasing it back out into the fluid to then interact with these microglia. So you have this, it's like they're constantly relaxing the microglia. But if something bad happens, then you suddenly have this stopping of the production of this fractalkine or the cleavage of this fractalkine. And then that means the microglia are now not in a dormant state because they're not being bound by fractalkine. So that's kind of the overall idea, that it acts as a constant off switch for the microglial cells as it's secreted from these neuronal cells. And that's been shown in vitro studies, meaning that it's been shown in cell-based studies. So kind of just looking at a plate and adding fractalkine and then seeing what kind of effect it has. And they've shown that also in more animal studies as well. But they've also had conflicting results when it came to the animal studies. So there's still some more research, considerably more research that needs to be done on this particular topic. But not only does fractalkine act as kind of an off switch to make sure that the microglia aren't being overly active. You want them to be somewhat active because you want them to be able to deal with whatever invasion might be occurring or what kind of issue might be occurring. But you also don't want them to be overly zealous. So not only does it do that, it can also act as an off switch, and it can also lead to this excitation or this release of what's known as adenosine. So not only is there an off switch for the activation of microglia, but microglia bound by fractalkine leads to a release of more adenosine, which counteracts the excitotoxic effects of glutamate by binding adenosine receptors, which downregulate the signaling of glutamate receptors. So it also leads to more gene expression of particular antioxidant molecules, and as well as the production of other factors like BDNF, so brain-derived neurotrophic factor, are released as well, which has been implicated in all kinds of different positive effects for the brain. So fractalkine not only pushes down or quiets down the activated microglia, but it actually also convinces these microglia to release more adenosine. And by doing so, that can then downregulate the activity of glutamate receptors. So if you have glutamate that's constantly binding, let's say, the neurons, what ends up happening is these adenosine molecules will bind adenosine receptors and then will feed back against the glutamate receptors and downregulate whatever downstream signaling is occurring by glutamate. So again, glutamate can be too much excitation, overexcitation, leading to essentially excitation toxicity. So adenosine has the opposite role, and it controls that. Now, not only that, glutamate can also increase the levels of reactive oxygen species. It can affect the mitochondria, leading to greater levels of reactive oxygen species. So the adenosine receptors can lead to a slight bump in more antioxidants being produced, which would help get rid of those reactive oxygen species. So fractalkine has a twofold effect, and actually has more effects than that as well, and I'll describe one more in just a second. But the point is that fractalkine doesn't have just this one effect and that's it. Just like a lot of things in the body, typically you maybe have a few main effects, and then you typically have a few kind of side effects that can happen as well. So that's two of the mechanisms by which fractalkine affects neuroinflammation. Also, it increases the expression of another protein, another neuropeptide known as MFG-E8, which is a shortened version of milk fat globule EGF8. And EGF is actually a really common protein that's released by the cells in general, but this particular one binds to particular receptors. I'm not going to go over the name of the receptors because it doesn't really matter, but ends up affecting these particular receptors that are a type of serine receptor, and those are on injured neurons. So on injured neurons, it ends up leading to an increase in microglial phagocytosis. What does that mean? Well, although it dampens the effect on microglia, what it also does is it encourages whatever microglia do get activated to then go in and clear out the debris of damaged neurons that are dying, convinces them essentially to end up undergoing what's known as apoptosis, or some form of cell death. So if you have this level of cell death, then you don't want a bunch of debris just kind of hanging out, floating around. That typically ends up overactivating immune cells, and then you have a lot of inflammation as a result of the debris itself. So if you can do that in a controlled way and kind of encourage the gobbling up, the consumption of that debris, the clearance of that debris, which is phagocytosis, then that's usually seen as a better outcome for specifically longer forms of inflammation. So you kind of have an acute form of inflammation where you have a small rise in the inflammatory markers and in microglial activation, and then it tends to die out. That's what you want typically. Now, that of course can be prolonged if you don't end up clearing out that debris. So that ends up leading to long-term inflammation. So fractalkine not only kind of modulates and acts as kind of a conductor for microglia in an acute sense in a short period of time, but also has some level of control over long-term inflammation indirectly. Okay, and that's through, again, that protein MFG E8. Now, here's something really cool as well, is that injections, and this is obviously not in humans, this is in animals. In animal models, they've shown that when they induce brain damage, that obviously fractalkine levels disappear or they reduce significantly. So what happens then is they inject fractalkine into the brain, and it seems like those localized sites of damage become a lot better, so that there's less inflammation, that there's less kind of secondary damage that ends up occurring at those particular sites. And preliminary evidence, and this is what I was alluding to earlier, that preliminary evidence of fractalkine reduces brain damage not only in localized sites, but there's also preliminary evidence that shows it has a protective effect in Alzheimer's, encephalomyelitis, which is inflammatory as well, Parkinson's, as well as epilepsy. So a series of really central nervous system specific and central nervous system related diseases are potentially positively affected by fractalkine's presence as well. So that's a pretty big deal. Okay, so now I'd like to kind of switch gears. I'm going to talk about neurotoxicity just for a brief moment. Then I'm going to talk about the relationship to humans to make this a little bit more applicable, and then we're going to finish this podcast up. So neurotoxicity of fractalkine, like I mentioned, not every study has shown that fractalkine is highly beneficial and is like this wonder molecule that affects the brain in so many great ways. If you were to look at it that way, that would be potentially a bit naive based off of the research that I'll go over now. So knockout studies, meaning that they genetically knock out a particular receptor, or they knock out fractalkine itself so that the cells can't express it or can't express the receptor. In this case, they knocked out the fractalkine receptor on microglia specifically, and it seems to reduce the damage from ischemia. So you would think that if you have this fractalkine receptor, that then you could have this action of fractalkine on the receptor, and therefore you reduce inflammation, therefore you reduce damage to the localized damage in general. But in this case, they're knocking out the receptor, and they're actually seeing reduced damage in local area, which means that you're getting better outcomes when you knock out the receptor. So that implies that there's also worsening ischemic damage, or a lack of blood flow to that area if you have fractalkine functioning, or more so the receptor. We can't really make any conclusions about fractalkine itself, but at least the receptor. So if the receptor is present, then presumably in this situation, they've shown that it can be damaging. So that goes against what I was just talking about previously. Another study showed that increasing the expression of fractalkine and its receptor in a particular area of the brain known as the substantia nigra, which is important for movement, so kind of just being able to move your arms, move your legs, etc. And then what they did is they with injections of fractalkine, so they artificially increase fractalkine levels, led to greater microglial activation. It's the exact opposite of what I was just talking about. So I'll speculate on this in just a second, but overall, they're seeing an increase they're adding fractalkine, and they're showing that that has negative effects on the microglia. It increases their activation. It also leads to dopaminergic cell death, so cells that are responsible for producing dopamine, as well as reduced motor ability dysfunction, which is very similar to what you would see with something like Parkinson's. So in this situation, high fractalkine levels was not protective, it was detrimental. And what's even more convincing about this is that it was reversed when they blocked fractalkine using an antibody. So they elevated fractalkine, saw the damage, then they put in an antibody that presumably, although this is sometimes speculative, and I probably should have looked into this a little bit more, it depends on how specific the antibody is, but let's assume, if we were to assume that the antibody is specific to fractalkine, then that binds fractalkine so that the effect of fractalkine is then removed, and they saw better outcomes once they blocked fractalkine. So there's some pretty damning evidence that fractalkine may not be that great, and it may actually be neurotoxic. So that obviously leads to a series of different questions, like why, right? Why is some research saying one thing, why is research saying something else? And that's where, as a physiologist, you end up having to dive into the literature and try to figure out, what's the context? What's kind of the driving things that are switching fractalkine's role in one situation and switching it in another situation? You can't. It's so dangerous then to look at molecules like that and just say, well, fractalkine is good for the brain. Done. We figured it out. It's never that simple. They're not usually that simple. So usually there's quite a bit of mystery that goes along with things. So what could be going on here? Well, it could be that it's very much dependent on the surrounding environment. So if there are other cytokines, if there's high levels of, let's say, interleukin-1 beta, which is the one I mentioned earlier, which is pro-inflammatory, interleukin-6, if there's high levels of tumor necrosis factor alpha, or on the opposite end spectrum, if you have interleukin-10, which is anti-inflammatory cytokine, there's a bunch of different factors. If the environment is potentially is overpowering where fractalkine can't have its effect, or if it has to have a combination of a series of different proteins that have to bind the receptors, or a series of different receptors on the cells to cause a particular effect. I mean, that's also possible. So you have to control for these kinds of things and see what else might be going on, especially if you're causing brain damage in a living system. That's something that, there's all kinds of things that end up changing. So it's possible that fractalkine can have a negative effect. Maybe it's up to a point, like it helps up to a point, and then after a point it has some sort of redundant mechanism that turns out to be a negative outcome for the brain. So honestly, you could speculate forever until you actually know more information on the topic. But the point is that context absolutely matters, which you'll hear from me many, many times in the future. Okay, so the relation to humans, well, patients with, this is more research that's been done, patients with bacterial and viral meningitis, which is an incredibly serious condition, which again is related to inflammation, as well as traumatic brain injury, which is what I've been talking about throughout, had elevated levels of soluble fractalkine in their cerebral spinal fluid, which actually goes against the point that the doctor said on the Joe Rogan podcast. So I'd be curious on why that might be the case, why there's a difference there. Maybe it's because it's meningitis, or maybe it's the type of traumatic brain injury. Maybe it's the site of traumatic brain injury. I mean, there's all kinds of different things that you have to consider. So that certainly goes completely in the face of what I was talking about earlier. Elevated levels are associated with better patient outcomes, however. So in general, if you have higher levels of fractalkine, that is at least associated with better patient outcomes. And it seems that based on limited evidence, that fractalkine likely reduces the inflammatory response and improves outcomes in conditions like stroke. So again, a lot of conflicting information there. It seems like the most of the research points to it being beneficial, but there is certainly some things that need to be figured out. We can't just ignore the other research and just say, well, yeah, we're just going to turn a blind eye. I mean, that's called bias. You have to read that research and try to come up with some sort of explanation. You need to try to come to the truth in some way, which is something that hopefully in time I will have some time, depending on how interested people are in this, to actually be able to dive in and do some more research on the topic. Okay, so in conclusion, and the kind of the big takeaways from this, fractalkine is a molecule that seems to have a neuroprotective benefit through a number of mechanisms, increases in adenosine, blocking or kind of lowering the activity of microglial cells is kind of two that I mentioned. It's cleaved from the surface of neurons, so it's a transmembrane protein, meaning it's anchored in the membrane and then it's cleaved by the proteases. Then it binds to microglia. It reduces inflammation in doing so when it binds. In most cases, now again, there were those exceptions. In some cases, it can worsen conditions, but it's speculated that it's based on the state and context or the environment of the microglia specifically, which are affected by fractalkine quite a bit. Fractalkine may also be beneficial for Alzheimer's, epilepsy, and a few other really major brain-related disorders. Okay, well, that's what I've got for you. Hopefully you found this informative, in terms of like what you can do to raise your fractalkine like that, or if you want to, like those are things that we're still going to have to do a lot more reading on and research, and hopefully we can come to some sort of conclusion on that front as well. But neuroinflammation is an area that's really starting to gain a lot of traction and its relationship to a series of different diseases. Unfortunately, this happens a lot in medicine. People find a link or a pretty strong link to a particular thing, let's say mitochondria or the endoplasmic reticulum or inflammation, and then they're like, well, all diseases are because of this. And that's almost never been the case. That has never been the case up to now. People, I think scientists just get excited about their field of research. More people get into it, and then they're like, yeah, it totally corroborates. There's this huge link. So therefore, it must be inflammation. That's the cause of Alzheimer's, the cause of Parkinson's, the cause of all these different diseases, ALS, whatever it might be. Usually it's not the case. But anyway, that's what I've got for you. We'll cover more on this topic if you're interested in learning more. And with that, I hope to have the pleasure of speaking with you in the next one. Have a good one, guys. See ya.